All right, here we go. Today we'll talk about vector competence in the tsetse fly. So as a review, what is vector competence? Help me out. For the few people who are listening in the chat, somebody says how good a vector is at spreading a pathogen. Yes, it's like the idea that you can quantify like the ability to spread a pathogen. And there's actually like a vectoral competence equation, but it's like the idea that you can quantify how good things are at transmitting pathogens, and not all insects are created equal in the ability to transmit a certain pathogen. So this lecture is essentially like a giant list of, like, if you could think of all the different mechanisms of things that might adjust or change vector competence, what would they be? What would be on that list? Okay, so that's kind of the gist of this lecture. So also just quick, in vector biology, we've seen the terms intrinsic and extrinsic in terms of like incubation periods. So the extrinsic one was what? The time that it takes for the pathogen to become transmittable in the vector. So that's like, in theory, like the time it takes for the pathogen to get out of the gut into the salivary glands and be spit out. Intrinsic is in the human. This is all review. Now, I'm going to use these words in a completely different context today. In a sense of what I'm going to do is I'm going to develop this list of intrinsic variables that are going to contribute to vector competence. And by that, I mean they're going to be like things like in the vector. And then I'm going to make a list of extrinsic things. So these would be like outside the vector, like environmental factors, things that are not in the vector. So intrinsic and extrinsic in respect to variables that change vector competence. So just briefly know I'm changing the context of these words, and we don't mean this today, we mean something different. Okay, so just brief review. Tsetse fly, males and females feed, they imbibe blood and pick up in that blood trypanosomes, which are the pathogen of human African trypanosomyosis and animal African trypanosomyosis. And there's a process that's like a chain of events. I've said this, I've made this analogy multiple times. There's a chain of events where the trypanosomes, first they have to do what? To get transmitted, they got to do what first? They got to get out of the gut. Very good. So one of these things is like, one, they got to get out of gut. And then they usually enter like the hemolymph. Then what happens? What do they got to do next? What? Well, okay, they're replicating, yeah, and they're going to replicate in different stages, and maybe there's going to be different like actual morphologies. But aside from replication, where do they got to go? What? Not the mouth. That's the filaria worms. Not in this case. That's in the kissing bugs. They get pooped out. These, in this case, they go to the salivary glands. Oh, I didn't hear that. Okay. Very good. Sorry. I partially deaf. Okay, yeah, they got to get to the salivary glands. Very good. And then they get spit out through the saliva. And that is one main mechanism of how trypanosomes and these diseases are spread by tsetse flies. Okay, and with this in context, let's also review the bottleneck hypothesis. The bottleneck hypothesis is the idea that like if you mess with certain links in this chain, you might be able to break the chain at the weakest link, right? So this relates to vector competence in a sense that, like, if all of a sudden a vector is unable to transmit a pathogen, there's been some kind of a break in the chain through multiple mechanisms, or one mechanism, or different types of mechanisms. You can get a break in this chain, and then if you put a break in this chain, that is effectively going to cause a different phenotype, which is the ability or not ability to transmit a certain pathogen. Okay, and now let's start talking about some of the variables that might be important for breaking this chain of transmission in the tsetse fly. Okay. So the first thing, intrinsic factor, innate immunity. Okay, so let's make a new one. Immunity, innate immunity. What does that mean? And this is in the context of it's in the tsetse fly.
Not learned immunity.
Yeah. Yeah, so like learned immunity in humans is like, oh, we get infected, we develop antibodies. That's like learned immunity. Innate immunity is usually like cellular mechanisms that are pre-programmed into the genetics that defend the system. Okay, and insects, like any organism, they have innate immunity systems which defend themselves against invasion. So if you're the tsetse fly and you're getting invaded by trypanosomes, like they often have cellular mechanisms. They don't want their whole body to be colonized by trypanosomes. Like that's not in their fitness interest. I mean, there's rare circumstances and nuances where maybe some pathogen might enhance the fitness of a vector, maybe under certain circumstances. But for the most part, like bodies do not want to be invaded by foreign bodies. Okay, so the tsetse fly and insects in general have innate immunity systems, and in general there's going to be like a push and pull, okay, of evolution, where the innate immunity of the tsetse fly is going to be evolving ways to kill or limit the replication of trypanosomes. And then the trypanosomes are going to be evolving like mechanisms to fight against that because they want to be spread. So there's going to be like a push and pull in terms of evolution that's going to cause changes in important things that relate to this. Okay. So just some questions, like let's just throw some ideas out there. Like what might change? What might adapt in these evolutionary scenarios?
Maybe like however they get out of the gut, something would like disrupt that.
Oh, wait, you're talking. Okay, I got to turn up the volume if you're talking. Say it again.
Maybe like if, I'm just guessing, but like if like whatever allows them to get out of the gut of the tsetse fly, something would stop that. Like that would be like a bottleneck like place. Like if it couldn't get out of the gut of the tsetse fly in the first place.
Yes. Okay, gut. Let me put that on the list of things. I'm going to put N here because I don't remember the number. Gut stuff. That's actually in the lecture later. Now I'm more thinking about like mutations in specific genes. Like what specific genes can you think of that might change, or like general concept families of genes? This should be kind of easy. Like what might change? Like things on the surface, right? Like there's going to be like surface protein adaptations that might change. Like Lauren said, things in the gut. So for instance, maybe like trypsin, gut proteases might adapt in a sense that they kill the trypanosomes. That could certainly happen. So these are just kind of examples. And these are examples of things that could change, actual genes, genetic genes that could change, that could then contribute to all of a sudden a change in vector competence. Does that make sense? You get the gist? Okay. Okay, let's talk about tsetse flies specifically. So tsetse flies have a lot of variation in vector competence. I'm just going to write VC, species to species. And this is one reason I like to use the tsetse flies as the example of this. They have a lot of variation in their ability to transmit trypanosomes. Okay, so let's take some different species. Glossinia morsitans. This is one species. Okay. It might only have, if you pull these out from the wild, it might only, I'm looking at my notes here. Let's say, here it is. Here's the number. You pull it out from the wild, 11% of this species have trypanosomes. Okay. And of that 11%, only, where is it? 20%. Only 20% of the 11%. So 20% of those 11% of flies that have trypanosomes, only 20% of those trypanosome infections get to salivary glands. So do you see again how there's like levels in this chain? Okay. So first chain is like, if you're going to spread this disease, you got to at least like have it in you. That's only 11% of Glossinia. Second piece of the chain is it's got to get out of the gut, get to the salivary glands. Only 20% of infected flies get to that point. So there's a term for this in trypanosomes and tsetse flies. It's called Maturation Index. I'll put this on the test. The Maturation Index. That's the term that means it's like, okay, what's the quantitative value of how many of these trypanosomes get from the gut to the salivary glands? That's the Maturation Index. So for Glossinia morsitans is 20%. So if you want to know, like, well, if I just pick a fly at random from the wild, like what's the percent chance that if I just let it bite me, I'm going to get, or it's going to be a fly that can transmit trypanosomes. Okay, so to get that, you take the 11%, so 0.11 times the 20%, 0.20, and that comes out to a 2.3% chance. Yeah, very good. To essentially be bitten by a fly that's ready to transmit trypanosomes. Okay, so this is an example. And now let's show you how they're different. So in a different species, Glossinia, I have it in my PowerPoint. I think it's Pallidipes. I don't want to waste time. I think it's, fact check me if you're watching online, fact check me. I think the species is Pallidipes, something like that. Okay, fact check me. But it's a different species. It's not morsitans. And in this one, okay, I'm looking at it. Only if you pull these ones from the wild, 1.3% are infected. So that's significantly different from this one, which is 11%. Okay, in this case, it's 1.3%. And the maturation index is 88%. So in this case, in this fly, it's like completely different. In this fly, it's like they're more likely to have it in their guts, okay? But they're less likely for it to mature to the salivary glands. In this fly, they're far more unlikely to have it in the gut, have it in them. But if it's there, 88% of them, a very, very high rate, make it to salivary glands. Okay, so the maturation rate or index here is 88%. So to get this, the same, if you calculate the same probability, like if I get bit by this fly, what's the probability that it is able to transmit trypanosomes? It would be 0.013 times 0.88, and it actually comes out to a 1.1% chance. Okay, so they're different. This one versus this one, it's different. That's just like the gist. Like the vector competence is different, and it has to do with maturation index. It has to do with what percentage like of the blood meals that they're taking. Are they picking up infected blood with trypanosomes? And there's more things, that variables that go into this equation. Okay, it's complicated. But these are two examples of how vector competence can be completely different in a slightly different species of tsetse fly. So you have to, the gist is you have to take the whole chain of events into consideration when you're thinking about vector competence. And there's more events that we're not even discussing. So how would this relate to vector control? So if you're like a public health worker and you have to be in charge of taking care of the tsetse fly problem, should you spend more of your time focused on Glossinia morsitans or more of your time focused on Glossinia pallidipes? Yeah, you probably want to spend more of your resources focusing on Glossinia morsitans because it's essentially like double. It's essentially like double the ability to transmit. So if you have to make a choice, I mean, maybe whatever your vector control regimen is, maybe it is equally effective at killing both of these, so you don't have to worry about it. But if there's ever a scenario where you have to choose, you probably want to look at the vector competence of either species and make an appropriate scientific decision on where to spend your resources if there's ever a scenario like that. Does that make sense? Okay. So another gist, let's go back to our original list. Things that can be different that affect vector competence. So there can be genetic differences. So let's talk about that. So these would be like SNPs, differences, different alleles in the genomes that actually contribute to differences, okay? We want to know like what these are. So there's a lot of people who study the genetic differences that lead to differences in vector competence. There's a lot of people who study these because these are important genes, important mutations, and they can be important for monitoring. If you want to look at a particular, if you're concerned about one particular SNP, which makes a mosquito or tsetse fly much worse, you might want to monitor that gene and where it is, where it's recombining in the wild. Another interesting fact that relates to this, that the idea that vector competence is controlled by genes, that means that vector competence is inherited. So that's why we talked about in the last lecture, one of the things we worry about is sex and recombination of these different strains of, in that case, we were talking about the pathogen, so it's not necessarily vector competence. But these are things that we worry about. Like these are inherited, so we worry about these traits being selected on by natural selection, and we worried about these traits being spreading or being passed on and spreading into populations that overlap with humans. Okay, so let's do a quick review. Let's talk about the different trypanosomes. So there's Trypanosoma brucei gambiense. What was the gist of that one? Good review here.
It's not the worst.
It's not the worst. You're right. This one will, but it'll infect you for years. So it'll be like a 4-year infection, but the death rate is much lower. Okay. And then this is we're talking, what disease are we talking about here with this one? We're talking about human African trypanosomiasis, sleeping sickness. Just to be clear, not Chagas disease and not Anagana, animal. Okay, yep. And then there's T. brucei rhodiense. Okay, and that one was the worst one. It infects the CNS. It'll kill you in, I think I said like 3 to 4 months, maybe a month, something like that. And this one is also causing human African trypanosomyosis. And then there was T. brucei, which was the animal African trypanosomyosis, the Nagana. And this isn't necessarily vector competence, but what I want to point out here is that these pathogens are different. Okay, they're different strains, they have different genomes, they have different SNPs, and we talked about a specific SNP, or we talked, I can't remember what the gene was, but we talked about that the rhodiense strain had some special gene that it was a causative factor for one of the reasons why it was worse. Okay, and so it's not just the tsetse fly that determines the vector competence. Okay, it's also the pathogens. A particular pathogen might have a gene that makes its maturation index. It might adapt its maturation index, making it more likely to mature. Okay, so in terms of like extrinsic factors, I think we could say, I mean, it's technically in the tsetse fly, so we could parse hairs about where this technically goes, but it's not in the tsetse fly genome. So I would say this is kind of extrinsic in a sense that the pathogen genotype is going to have an impact on vector competence. Okay. So in humans, more interesting examples. And Dee, you kind of actually asked about this. I think you said something about animals might develop some resistance. So in humans, there's a gene called TLF. It was given a name after it was determined to be a trypanosome lytic factor. So its name stands for trypanosome lytic factor. Okay, perhaps it does other things, but in many cases, proteins are sort of named after the first function that they've been characterized to have. Okay, and so there's this gene. What I'm telling you is there's this gene in humans. It kills trypanosomes. Okay, so what it is it's a lipoprotein. Where do you find lipoproteins? This is a human cell. What? I can't hear you. In fat. Well, fat is a very okay, and you're thinking, because your background, you're thinking in a very organismal sense. Think in a cellular sense. Where are you going to find a lipoprotein? Well,
You can find it on the membrane, like the cell membrane.
Yes, you're going to find it in the membrane. Lipo you're right in a sense that lipo means fat. It's essentially, but the lipid bilayer is made of fats, right? So you're going to find a lipoprotein in the bilayer, and these things usually expand the bilayer, and sometimes they stick out, and sometimes they have interesting things that stick out of the bilayer. Okay, so you're going to find this trypanosome lytic factor in the bilayer. And what happens is, let's get a different color here. If you are a trypanosome, so right, they have that flagella thing and the kinetoplastid. If you are a trypanosome, and they see this, okay, they touch this thing, and they take it into their system, and it kills them. So there's like a cellular defense protein that kills trypanosomes if they try to eat it. Okay. But this only kills them if the trypanosome is T. brucei gambiense. So maybe another reason why brucei gambiense is not so bad is because humans have actually evolved like a protein that kills it. So we have resistance, in a sense, against that strain. Okay. But in the case of, let's get a different color. In the case of T. brucei rhodiense, Rhodiense has a special protein called SRA. SRA is, I think it's a, oh yeah, there it is. It's a glycoprotein. What's a glycoprotein? What's a
A sugar.
Basically, has a sugar attachment.
Sugar attachments, and oftentimes you find things that are glycosylated also on the surface. So you will find important glycosylation signals on the surface. So Trypanosoma brucei rhodiense expresses this interesting protein called SRA, and it's a glycoprotein. And this protein, it essentially, it will, what do you call it? It'll inhibit or counteract TLF. That's an F. So this adaptation that we have, it can't kill Trypanosoma brucei rhodiense because the Trypanosoma brucei rhodiense has also evolved a counter mechanism that inhibits the TLF. Okay, so do you see how this is like an evolutionary arms race, and some strains are more ahead and more advanced than others? Or you could say maybe perhaps Brucei gambiense is attenuated in a sense that maybe it's learned to, maybe it's just sort of learned to get along to go along. Whereas Brucei rhodiense has evolved a strategy to inhibit this thing, and it's much more virulent. Okay, so this is an example. I give you this as this is a specific studied example of this evolutionary arms race happening. And in this case, it's again, it's not, maybe not vector competence in the sense that it's this is taking place in the human, but you could envision a similar scenario happening in the tsetse fly, and that would impact vector competence. In this case, it's actually a molecular mechanism, a single molecular interaction that's determining whether or not people live or die if they get trypanosome diseases. So I think that's pretty interesting. Let me skip that. Or maybe I will. How would we, let's go into this a little bit more. How would we study this and prove this? Like if say I just told you about this and now I say prove it in the lab. How would you approach that? Experimentally, how would you prove this to me? What are some ideas? Hold that thought. Let me just start off that the first thing you would do to even like discover that this might be something of interest is, and we talked about this, you would sequence both strains, one that's more virulent than the other. You'd sequence both the genomes, and then you would compare the genomes and you'd look to see, well, this one has something really unique. And they probably determined, well, this one has this SRA protein. That's what makes it unique. So now let's study the function of SRA. Okay, now give me your thoughts. So now the question is, okay, let's study the function of SRA. Prove it to me that it's doing this. What would you do? Yeah, you can do a knockout. You could do a knockout.
Knock off the SRA gene and see the effect, and then compare it with when the gene is on.
Yeah, so you could knock it out, and you could test invasion. You could measure invasion in like cell cultures. You could measure pathogenicity. I don't know. It'd probably be in maybe like an animal model. You wouldn't be able to do it in humans. But this is definitely one thing that you could do. How do you knock out the gene? That's a different class. It depends on what kind of an organism it is, and it depends on what tools you have available. Just quickly, if your question is like, how would you knock out a gene? You could use a transposon. It would be random though. You'd have to get lucky. You could use an integrase or a recombinase if it had homologous recombination. You could use CRISPR Cas9 nucleases, and you could cut it, and in theory, it would get mutated. You could, how else could you knock it out? These are typically the most common ways people are knocking stuff out. And if you're interested in that, you should watch my biotechnology lectures. But that's a subject for a different class. Did I miss any mechanisms of knockout, Lauren? I don't think I did. Okay. Mute that microphone. Getting strange noises. Thank you. Okay, let's go back to our list. Other things. What about sex? And by that I mean male versus female. So I don't know if that's gender or what, but sex. How could that cause a difference in vector competence?
Because if something's carried on like the X chromosome or the Y chromosome, females would have a double chance of getting it. Or if it's like just on the Y chromosome, then maybe only like males could get it.
Yes, that was exactly my point. Is literally in many cases, even in insects, in humans for sure, and in most insects, what conveys sex differences is in the case of well, okay, insect sex differentiation gets complicated, but many systems are XY, XX, just like we are. Okay, and so the gist is that there is a sex chromosome, and if there's some gene that's sex linked, if there's some gene, like maybe for imagine, I mean, this is not the case, but if maybe, for example, that SRA gene or some other gene that was important for interactions in the tsetse fly, if that was on the Y chromosome, the males would clearly have a different transmittability, a different vector competence than the females. So, and this is actually like a thing at the NIH is, you have to treat the sexes differently. They actually like explicitly say this to you when you write grants. That's actually one of the criteria. Have they considered sex as a variable? And have you thought about the cases in which males can be different than females? And are you controlling for that in your experiments? So that's an important thing that could play out and cause differences in vector competence between males and females. Okay. So obviously, many mutations could occur. And we talked about things like outer surface proteins would be important. Variable surface proteins could incur mutations, glycoproteins, gut proteases. I'm just kind of ranting off the list that we've talked about. Gut proteases. Many of these things are going to be accumulating mutations and causing changes in vector competence. But these are all specifically changes in an ORF, the open reading frames that are genes that code for proteins. Where else could you get mutations that might change vector competence? So could mutations occur anywhere else, not just in genes, that could cause changes in vector competence? Yes, exactly. Like regulatory elements. So you might not actually have a SNP difference between ORF1 and the allele in a different strain, but you could get a mutation in the promoter, which causes it to either be upregulated or downregulated. And then now all of a sudden you have a huge difference in vector competence, but you don't have any difference in the actual genotype. Does that make sense? I mean, I guess you have a difference in the genotype, but you see the nuance here that I'm talking about. You could also have things in the terminator. You could have things in the 3' UTR that is important for localization of transcripts. You could have possibly mutations in introns that are important for splicing. You could have mutations in enhancer regions upstream. You could have mutations in transcription factors, which it's actually not the transcription factor that's conveying the difference, but perhaps then that transcription factor is not expressing a certain ORF downstream as much, and so then you could get changes. So this is just, I'm just showing you that it's not just changes in genes that can cause changes in vector competence. It's also just sort of any kind of change on the chromosome might have an impact. Okay. I don't know if that's worth going into. Let's talk about the gut. So in the gut of the insect, what's that environment like in guts? What are guts famous for? Yes, digestion. So there's going to be a lot of things that contribute to digestion. Okay, there's going to be proteases. We mentioned trypsin. What else is going to be in the gut? Or like what conditions are important in the gut? Yeah, the pH. That's a famous example of gut stuff. Is when you go into guts, insect guts, human guts, animal guts, the pH changes. It's not always that it gets acidic. Sometimes it gets basic, but the pH has drastic changes. Why would pH potentially change things in a pathogen? So if you're this trypanosome inside the gut, what's actually going to potentially be an issue for you if the pH changes? Like what is physically actually the mechanism that's happening? Yeah, so let's talk about that more. Okay, so that might be a good example. Your outsides are going to be, or your membrane or your cell wall, maybe not a cell wall in this case, but your membrane is going to be made of lipids, but also proteins that sort of sit in that membrane. And proteins are made of amino acids, AA. And certain amino acids are susceptible to pH, right? Like certain amino acids actually change kind of like conformation, chemical composition during different pHs. Some amino acids are basic, some amino acids are acidic. Under certain conditions, certain amino acids become positive or negative, okay? So when you go all of a sudden put a protein in the gut, it's not guaranteed that it's going to hold that conformation that it's used to. So if there's a gut pH situation that starts to change things, that can affect the vector competence. So imagine you have a horribly acidic tsetse fly gut that is like really good at just like destroying trypanosomes. It's going to be low vector competence. If there's a scenario where the gut pH is maybe not as acidic or not as basic, maybe it doesn't have as much of an effect. So it has high vector competence and they can replicate in the gut and then they break out of the gut, okay? Or maybe there's situations where the trypanosome has evolved its outer surface protein to be perfectly adapted for the gut pH of the tsetse fly. And then maybe in that case, it's high vector competence because it's really evolved for this particular gut niche, okay? So these are just examples. I'm not giving you specific. I'm not saying this is exactly how it happens in Brucei rhodiense in tsetse fly. I'm saying these are examples. These things can happen. Let's talk more about, so, okay, did I get that? Guts, okay, yeah. Let's talk more about the insect innate immunity systems. You're gonna hear, if you actually read about like insect immunity, you're gonna see things like toll pathway, IMD pathway. These are cell signaling mechanisms. I don't expect you guys to know these. And even if you look at like these like biochemical pathways, it's just essentially like a big list of, oh, this does this, and then this signals that, and then this signals that, and then this signals that. It's like, it's not useful to know unless you like really study it. Just know that like there's a thing called the toll pathway and the IMD pathway. These are cell signaling pathways, and they're often important for innate immunity. So they cause things to be upregulated that can help the insect fight off pathogens. Okay. There's also, you do want to know this, because this is very important in insect biology, comes up all the time. there is a type of molecule, a type of protein that insects make called AMPs. What do those stand for? Do you know? It's anti-microbial peptides. Yeah. So there's actually genes in the insect chromosome, and they code for antimicrobial peptides. So they're little peptides. And what these peptides do is, if there's something like a trypanosome or something like a single-celled organism, in the hemolymph, the insect will secrete these AMPs, and the AMPs are essentially, they essentially form pores in the membranes of single-celled organisms. And then what happens when you form a pore? The organism pops. So they're essentially proteins whose mechanism is to go across the membrane and pop bad guys. Okay, so insects have these. One specific example is called cercropin, and we'll read a paper specifically on cercropin in either the next lecture or the lecture after that. So if you have a tsetse fly that has a particularly good repertoire of AMPs, they might have low vector competence. Okay. Insects also have what are called hemocytes, which are what? Where do hemocytes live? They're cells. Where do they live? In the hemolymph. Yeah, in the hemolymph. And they're essentially like they're like white blood cells. Like they will go and they will find bad things that are floating in the hemolymph, and they will surround them and kill them. Okay, so insects have hemocytes. If the tsetse fly has particularly good hemocytes, it's going to have low vector competence. Okay. More things. Oh, let's go back to our original list. This could be this is more again, this is debatable whether it's intrinsic or extrinsic, because it's inside of the tsetse fly, but it's not inside the tsetse fly genome. But symbionts. Okay, and I should talk about this more, but I don't have enough time. But tsetse flies have 3 symbionts, and there's a lot of research done into how these symbionts affect biology of trypanosomes. Okay, and 2 of these are gut symbionts. So one of these is Sodalis, which is a bacteria. Another is Wigglesworthia, which is a bacteria. These are the gut symbionts. And then there's a reproductive symbiont, Wolbachia. And there's a lot of effort been studied into, because Sodalis and Wigglesworthia, they're going to be in that gut with the trypanosome. And they want to live there. These are like sort of like healthy tsetse fly symbionts. So they don't want their host to be like infected by this foreign pathogen. And so there's potential pathways where a Sodalis might do something to inhibit or kill the trypanosome, or a Wigglesworthia might do something to inhibit or kill the trypanosome. Okay, I'm not going to go into specific examples, but just the general idea is like there's other things in that gut that might kill the trypanosome, irrespective of the tsetse fly genome. Okay, let's talk about other environmental things. I have a whole bunch of these slides, like a whole bunch of things. These are all more extrinsic. So let's start making a list of extrinsic things. Let's talk about like weather. How could weather affect vector competence? So the climatic conditions under which certain strains live, how could that impact vector competence? You have to think creatively for this.
I mean, this isn't really with tsetse. Oh, is someone else talking?
No, it's just super loud. Okay, go again.
Oh, I'm sorry. Like kind of not, I don't know if this is with tsetse flies, but like with the mosquitoes, if there's not like water for them to have their eggs in, like they can't lay eggs. So like I know like for one means of their vector control, they were saying like get rid of all the things that would catch water if it rains. So like ways to interrupt like I guess their life cycle, but I don't know if that.
Well, you're close. You're close to one thing I'm thinking about, which when you talk about like water. Okay, when you think about the African savanna, where these tsetse flies are living, like they're famous for these like water holes. Otherwise, in many cases, it's kind of like a dry, arid place. Okay. And so essentially, like, weather patterns are going to affect the geography of like where the animals are, where the tsetse flies are, because it's going to impact when things are going to the water holes, the daily patterns. Like if it's too hot, nothing's going to be moving. The tsetse fly is not going to be moving, but maybe there's a case where, so what I want to say is weather patterns could produce a mismatch between tsetse fly behavior and its blood source behavior. Or it could cause a scenario where they now line up. And the scenario where they line up is like, oh, it's hot. Everything goes to the water hole. Now everything is there. The tsetse flies are there. The reservoir is there. And it's picking up a blood meal. In that case, it could increase the vector competence. Okay. They're floating around in, so remember like Nagana, that's floating around like in the animals and in the cows. In terms of human African trypanosomiasis, it's in humans. I don't know if there's a sylvatic cycle. So I don't know if there is a human or if there's a non-human reservoir for Trypanosoma brucei gambiense, but there must be. I should have that in here. Somebody fact check me, but I can't give it to you right now. Probably some ungulate. Host preference. We talked about this a lot in the context of mosquitoes. So something is not going to be a vector for humans if it only drinks bird blood. If it never bites humans because it's just not interested, it's not going to vector any diseases. Okay, so that could be an extrinsic factor, a behavioral factor that impacts vector competence. And then I would also just kind of list, as we've talked about, geographical overlap. Like your reservoir has to overlap with your vector, has to overlap with humans if there's going to be transmission of a zoonotic disease. Let me just check this PowerPoint to make sure I didn't miss anything. Blood source. This kind of relates to Dee's question. I guess goat and pig blood promote higher midgut infections of trypanosomes. Age. This is very important for mosquitoes, right? Like the age of a mosquito is going to change old mosquitoes that are transmitting diseases. It's not. Nice. Yes. So age can cause a factor. Nutrition. So how well has that tsetse fly been fed? If it's been fed really well, maybe it's really robust. Its immune system is real good and can fight off the trypanosomes. Or maybe it's the opposite. Okay. So these are all just kind of examples of all the things that can impact vector competence in the specific instance of tsetse flies. Okay. So we'll end there. Let me end this quick. Oh wait, no, I don't want to. Oh shoot, it's not going to stop recording. The last thing I'll say is the next lecture will probably be the Chagas disease, or we'll read a paper for Chagas disease. That one's already done, so that will probably just be online, and then I'll keep you guys updated. So Friday will probably be an online class.